wherever you're watching me from. So once again, George's Solutions, and I want to welcome you to my YouTube channel. And before we start, like the norm, remember to click that subscribe button. So today we'll be doing something on viruses, and to be even more precisely, we'll be doing something on coronavirus, and to be even more precise, we'll be doing something on the newest variant called the Omicron. Characteristics of the viruses, how they replicate, and what a viral disease is. Then finally we will break it down to the Omicron variant that has been recently discovered, what we should do to stay safe, all those we will do. Viruses, let us go right away. Viruses, we consider viruses as obligate intracellular parasites. So when we say obligate, what do we mean? We mean that these are organisms or microorganisms that cannot survive on their own. They cannot individually survive on their own. It means they require a host cell, and a host cell can be me, it can be you, or it can be a plant or an animal. So they are not able to individually produce metabolic activities. They can't produce energy, they can't produce proteins by themselves, but they require a host cell for them to be able to survive. The replication is not the division, but they are simply of subparticles. It doesn't divide, but it requires assembling of subparticles for it to replicate. So the genome of viruses can either be an RNA or a DNA, but it can never be both. A virus can either be an RNA virus or a DNA virus, but it can never be both. And we will actually break it down to you later on and tell you why RNA viruses are more lethal than DNA viruses. So viruses, first of all, they are not living. They must be infectious to survive, and they must be able to use the host mechanisms to produce themselves and to produce structures like the messenger RNA, proteins, and copies of the genome. They require the host to produce all this. And also the subparticles must be able to assemble, and viruses are very, 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 very tiny. Viruses are classified according to the structure, shape, morphology, biochemical properties, the diseases they cause, how they are transmitted, their host cell, tissue tropism. There are a lot of ways in which we classify viruses. And just to tell you a brief history of how viruses were discovered, this is just a by the way, but a long time ago, people believed that all particles that cause diseases, let me just say pathogens, all pathogens that cause diseases could be filtered in the laboratory. They could be filtered in the laboratory, and when you are filtering it, you filter it, it means that it will remain as the residue and there will be pure water at the end. But there's this leaf mosaic virus that was filtered, and when they took the water after filtration and infected it in a healthy tobacco plant, the tobacco mosaic virus still infected the healthy plant even after filtration. So this later on is what was discovered as these viruses, and they used to call it vividum fluidum, something like that. Protein in nature, and they can spread through stool, dirty hand, dust, small droplets, and even after drying, some of them are infectious. They are able to survive in unfavorable conditions such as the colon. They resist detergents, and they also stimulate the production of antibodies. So according to the structure of the virion particles, actually a single virus is called a virion particles. is called a virant particle, and according to the structure classification, it can be a viron or a nucleic acid. That one is the one I said. It can be RNA or a DNA, and when it is RNA or a DNA, it can be single-stranded or double-stranded. It can be segmented or non-segmented. It can be linear or circular. It can be symmetrical, icosahedral, helical, or complex. It can be encapsulated or non-encapsulated also. You also classify it according to the number of capsomeres. It can be non-conventional viruses. So all those are classification of these viruses. And actually types of DNA viruses, we have the herpes, papavirus, adenovirus. Those are all DNA viruses. We have like the corona, we have the HIV virus. All those are RNA virus. So let us go through the replication stage, the replication of viruses. So first of all, for a virus to replicate, there must be something called recognition of target cells. So the virus, first of all, must recognize a target cell, and this one explains even why HIV only goes for the immune cells, and the coronavirus also only goes for the respiratory cells. So it must recognize a target cell. It must have a target. It must have a target cell. After it has located a target cell, the virus particle will attach to it. After it has attached, it will now penetrate the cell. After penetration, it will uncoat. Then after uncoating, then there is what we call synthesis. At this stage is whereby the early messenger RNA and non-structural proteins replicate to form a genome, and also after forming a genome, they also replicate to form proteins. And after replication, there is the final stage where we call assembly. Now after replication, the parts come together to assemble together. They bud, and like in the budding of non-capsulated viruses, and then it is released from the cell. So I'm going to speak on something on genetics of viruses, and this actually forms the backbone of our discussion today. So the genetics of viruses, we'll majorly speak of mutation. I know you have heard of mutation before, and if you haven't heard of mutation, don't worry, because I got you sorted. So mutation can generally be defined as changes of characteristics of a daughter virus in comparison to the wild type. So the wild type is the original type of the virus. So the daughter viruses that occur after replication are now having specific changes. This is what we call mutation. So mutation can be of general genes, the inactivation of viruses, and this can lead to lethal mutations, and mutation of other genes can lead to changes in the properties. This can involve the deletion, attenuation of properties, changes in the host or the target tissues, and resistant to temperature. All these can be aspects of mutation. And sometimes also mutations can be induced chemically, such as by radiation. When you are radiated, this can lead to mutation of the virus, or by nature, caused by insufficiency of the viral polymerase. The polymerase enzyme is the enzyme that leads to the production of other viral particles. So when the polymerase is insufficient, this can lead to mutation. And mutation is more common in RNA viruses than in DNA viruses, since when an RNA virus, it needs to actually replicate to form the DNA form during its transmission. So that is why in this process of replication, there will be some errors that will be accrued, and this can lead to mutation. So there's also what we call the recombination. So recombination is the co-infection of two similar viruses. So we have two similar viruses and they can recombine. So the viruses with segmented genomes are assemble in two different defective viruses. So when they reassemble, we call this recombination. So next thing is that I'll speak of something on viral diseases. And before I continue, I hope you are still with me. And if you haven't subscribed to my channel up to this far, I'd still just ask you to please subscribe to my channel. So right away, let's go to the viral diseases. So the virus itself can avoid the immunity, or it can take control of the immunity itself by killing of important cells or production of destructive immunity or inflammatory reactions. So immunity reaction is actually one of the best therapies for viral infection, and it is also the most powerful factor of pathogenesis of a viral infection. So it is your immunity that will help you to fight this virus. But some viruses have mechanisms in which they can actually evade this immune response and actually cause immune responses that are unfavorable to your body, and this is what we call a viral disease. And also there is what we call tissue tropismus. Hey, this one is a hard one. There's what we call tissue tropismus, whereby different diseases can be caused by the same virus. You get that you have one virus, but you suffer from different diseases as a result of this same one virus. So one virus can produce different diseases. So infection can occur by entering to an organism through the skin or the mucous membrane, protected by the tears, epithelium, or stomach acid. We can also inhale most of these viruses, like specifically the coronavirus. We inhale it, and it is the most common way of transmission of infection. The inhalation is the most common way of transmission of an infection. So let us get to the core of this session. So on 26th of November 2021, the WHO realized that there is a new variant of concern of the COVID-19, and it named it the Omicron variant. And all this happened due to substantial evidence of how it mutates, or how it spreads, or how it generally causes severity of illness. And I want to break it down to you, a summary of what as at today is currently known about the new variant. So the origin is in South Africa, and around the world they are still conducting a lot of studies to better understand the many aspects of this Omicron variant. And as time goes, we will keep sharing more and more information about what has been developed. So first of all, let us speak of the transmittability. And I have to say that it is not yet very clear of how the Omicron is more transmittable. For example, how it easily spreads from one person to another compared to other variants, including the Delta variant. This is still not very clear, because the number of people testing positive has highly risen in areas of South Africa, and these areas of South Africa has been greatly affected by this variant. So it is not very clear of how it transmits so fast, but epidemiologic studies are underway to understand this and many other factors. Also the severity of the disease. I'll tell you something on severity of the disease, and I can also say that it is still not clear whether infection with Omicron causes more severe disease compared to infections with other variants, including the Delta hospitalization in South Africa. But these results is not linked to the specificity of the Omicron variant. There is currently no information suggesting that symptoms associated with Omicron are different from those of other variants. There is no data suggesting that. Initially reported infections were among some university students, younger individuals who tend to have more mild diseases, but understanding the level of severity in this Omicron variant will take about several weeks for us to fully understand how severity the diseases, what it can cause. But it is also very, very important to remember that all variants of COVID-19, including the Delta one, can cause severe diseases to individuals, including even death, and hence protection and prevention of the virus is the key aspect to look at here. So reinfection, I'll speak about reinfection. For example, let's say you already had coronavirus before and now there is a new variant. So there is not still much data to tell whether there is high possibility of reinfection, though a lot of data is suggesting that reinfection with the Omicron variant will be high compared to other variants that have been presented before. But all this will take time, and more information about reinfection will come in the next weeks or days to confirm the same. I'll speak of how to test the virus and effectiveness of the current test that is available. So the current test that is available is just the PCR, which is the polymerase chain reaction or also called the nucleic amplification test. This is the most effective test for COVID-19, including all other variants, but there is a lot of research still going on a rapid antigen to detect this new variant. So I'll speak of the effectiveness of the current treatment available. So as at now, corticosteroids will still be highly effective to be used to manage people with severe coronavirus, even in this new Omicron variant, and the IL-6 receptor blockers will still be used to manage patients with severity. But more data, as I said before, more data will come to see if it is still effective, given that there are changes to parts of the virus in the Omicron variant. So what I can simply say is that studies are still underway. At the present time, the WHO is working around the clock, and there are a number of researchers around the world. They are still trying to understand this new virus, the Omicron variant. So we just need to give them more time. But the recommended action is that we just still prevent. We try as much as possible to stay safe, since prevention is always the best option than cure. We stay safe, we wear our mask, we wash our hands, we avoid crowded places, we practice social distancing, all those things for us to stay safe as we try to better understand the new variant of COVID-19. It is also very important to increase access to the vaccine. The vaccine have proved to be very, very good. COVID-19 vaccine are currently being very, very useful, especially to the vulnerable groups, including the health workers like me. We should go for the vaccines for us to stay immune.